Okay, I'm just going to do a quick recap because there were two papers that have come to my attention, and they're not actually even that old. So, or sorry, they're not even that recent. I just completely missed them, which is surprising. So the first one is one called, excuse me, pulmonary and systemic involvement of COVID-19 assessed by ultrasound. So this group down here in Brazil, yeah, Sao Paulo, Brazil, they are uh, to discover a non-invasive treatment of a way to look at or to do autopsies, because remember the corpses of a lot of people that die right now are contagious. They can still release a viable virus to the morgue workers. So what they were looking at here was what are some ultrasound methods in order to not only prevent the people from having to expose themselves to the corpses, but also what's some way that we can use ultrasound to verify whether or not people had contracted COVID-19 or contracted SARS-CoV-2. So here, how do I phrase that? I just want to bring attention to this and then also mention that we will find it, orchitis. So remember, orchitis is inflammation or swelling of the testicles. And so here they say when they went through this cohort of dead bodies, the main systemic findings were associated with comorbidities, age, and sepsis, in addition to possible tissue damage due to the viral infection, such as myositis, dermatitis, myocarditis, and orchitis. So this is something that I was pretty worried about from the very beginning. And this is an accepted paper, so it was accepted. And I am one of the reasons I'm concerned about this is because the testis uh, has very high expression of the ACE2 receptor, and we do know that with the other two serious coronaviruses, so SARS-CoV-1 and MERS, that there was orchitis associated with those events as well. Now, were the people who suffered from SARS and MERS, did they end up being contagious through their semen? It doesn't appear to be the case. Granted, with MERS, there wasn't a lot of person-to-person. What do you call it? There wasn't a lot of person-to-person transmission, but with SARS, it's unsure. I think a big problem was a lot of people ended up having their partners die from SARS if they survived. So I'm not sure how that played a role in sexual transmission later on. But here we can see that there is tissue damage in the testicles of the people who had died at least. And so one of my concerns is the presence of inflammation in the testicles. To me, indicates it is possible that viruses are getting into the testes and into the epididymis and other sexual organs, which would then result in um, possibly viable virus existing in the semen, especially and since it is such a long term, how do I phrase that? Since it is an immunoprivileged site, I fear that the virus will be able to exist in the semen long after someone has recovered. Okay, so this is my concern. So I'm not going to go over this paper. I just want to say that this was one of um, the conclusions that this work had demonstrated. So that's the first thing that I want to bring to everyone's attention. And then also I have it. I will leave the reference down in the description if you want to read this paper for yourself. And then the second one, this is coming out of Wuhan, China, and this actually came out a long time ago. This version was posted on March 30th, and I'm surprised that I couldn't find this paper before because I had been looking for papers like this, but it must have been in um, the titles of it that made it difficult to find. And keep in mind, I don't know if this ended up getting. I don't know if this was ever got past peer review. It might be in the process of peer review right now, but they thought it was important to release. But here it's the effect of SARS-CoV-2 infection upon male gonadal function, and this is a single center-based study. And one of the things that that really concerns me is remember I I will post it later, but I had a video going over some of the amino acid sequences that are on the surface of the SARS-CoV-2 spike. And I chose the SARS-CoV-2 spike to analyze the amino acid sequence because it's probably the protein that is most exposed to the immune system, and it's also produced in such high quantities that if you are going to mount an immune response, it'll probably be against those spike proteins. Now, as a my concern from that sequence was there was a section of the amino acid sequence, and there were quite a few, but one of them in particular was creepily similar to a different amino acid sequence of one of your peptide hormones. So it was the beginning part of the NPP peptide, which is a part of the POMC protein. And if these acronyms mean nothing to you, I have another video about it that I'll probably link to at the end of this video. And my concern was that this amino acid sequence has been studied before. It has been found in so peptides containing this sequence has been found in the hypothalami of pigs, and when they used those amino acid peptides on cultured mouse cells, they could see that these peptides had a function of inhibiting the release of prolactin, right? So whatever this peptide was, it was inhibiting prolactin release. So if you had a virus that was mimicking this particular amino acid sequence, then you might possibly start making antibodies against this sequence. And in fact, it's actually very likely. So one of my concerns was that if you start making antibodies against this peptide that exists in your body, then you will start sequestering that peptide, so the peptide won't be able to function. And then, as a result, you end up releasing more prolactin, and prolactin has a lot of downstream effects on your hormones as well. So imagine this with me. You start releasing a lot of prolactin, which could cause testosterone or other hormone imbalances to take place, or what was it? You have your, I mean, I'm going over this right now, but there you have your follicle stimulating hormone, you have your luteinizing hormone, you have all these hormones that are produced in the central nervous system that are tightly regulated, but are also in being regulated in part by prolactin. So if you have a large release of prolactin, you might start seeing gonadal effects. And lo and behold, what do we see here is that they use PRL. So here they tested a bunch of hormones. So testosterone, estradiol, progesterone, prolactin, luteinizing hormone, FSH, anti-Müllerian hormone. And let's just go down to, and they also had a control group. And so let's go down to, one second, political ads. Compared to the control group, COVID-19 patients had significantly higher serum luteinizing hormone and serum prolactin. And it was extremely significant. So here you see that the p-value is less than 0.0001 for both of them, right? And also, they said that there was no statistical difference in serum testosterone. So that's good, that at least the serum testosterone is the same. Or the follicle stimulating hormone, at least that was not statistically different. And I should mention, there was a difference, but it wasn't statistically different because usually you have to be, I shouldn't say usually, but the gold standard, if you will, is I shouldn't say gold standard. The standard is 0.05. So if it's above 0.05, they're not going to say that it's statistically significant. It is still possibly a result of random chance, right? Continuing, but what is statistically significant was the ratios of testosterone to luteinizing hormone and follicle stimulating hormone to luteinizing hormone. Because those were dramatically decreased in the COVID-19 group. So what I will look into, I'm not quite sure about this, is these ratios were probably important because these ratios probably play some physiological role in the body, which is why they were looking at them. Granted, since they know that luteinizing hormone was significantly higher in the COVID-19 group, we would assume that it would also be very likely that the ratio with luteinizing hormone in the denominator is those ratios would likely be very low if you're using the concentrations of testosterone and follicle stimulating hormone. Granted, there might be I'm ignorant on this subject, so I will look into it, but probably my guess is the ratios of follicle stimulating hormone to luteinizing hormone and testosterone to luteinizing hormone is also plays some physiological role in cellular signaling. So I will look into that. But it was significantly different. That's something that we really need to consider is there you have so much luteinizing hormone and so much prolactin that your hormones out of the COVID-19 patients are probably screwed up. So this is one of my worries that I brought up earlier was, is prolactin being released? And I hadn't read this paper before, and keep in mind it's not peer reviewed yet, but here is an example. If people actually look at the prolactin levels, there is a significant increase in the serum concentrations of prolactin. So what does this tell me? This tells me that we might be on the right track by looking at prolactin levels. And I should look to see if prolactin ends up causing the release of luteinizing hormone. But yeah, that's just all something that I really wanted to warn people about. So with that out of the way, I'm going to stop this, post it, and then I'm going to continue this paper, which was the testis and immune privilege, because we definitely need to finish the biology of the testicles if we want to learn more about how this disease could possibly be really negatively affecting the whole population, but then also how does it affect men specifically. So with that, thanks for watching, and I'll try to get the other video out by the end of the day as well. So thank you.